Hi guys, Miss Francis here to talk to you about non-cyclic photophosphorylation. I recommend that you guys revisit the McGraw-Hill animation. Just take a note that the McGraw-Hill animation starts with photosystem 2 and goes through the non-cyclic photophosphorylation, whereas we start with photosystem 1. Technically, both of these photosystems are used concurrently, but for us, we are going to start with photosystem 1 since we already know it. So before we dive into non-cyclic photophosphorylation, let's just take a look at where we are located. So photosynthesis, we know, occurs in the chloroplast. Now, we are still discussing the light reaction, so we're still talking about the thylakoid. One of the products that you're going to make from electrons becoming excited in photosystem 1 is going to be NADPH, and NADPH is going to be made in the stroma. So if we're looking at this image, we can draw a line here and say that this is the stroma. All right, so now let's dive into annotating the image. Once we're done annotating the image, I'm going to go back and address all the questions in the note packet. So we're going to start with photosystem 1. And what happens in photosystem 1 is that light hits photosystem 1, a primary acceptor accepts the electron, and an electron becomes excited. That electron then travels down the electron transport chain and reduces NADP plus to NADPH. So remember, NADPH is an electron carrier, just like NADH was an electron carrier for cellular respiration. But that electron, right, that was traveling down the electron transport chain didn't go back to photosystem 1. It was used to reduce NADP plus to NADPH. So what's going to replace that electron that's now missing from photosystem 1 is going to come from photosystem 2. So let's take a look at photosystem 2. Now, photosystem 2 is found in more evolved plants, and let's jump in. So photosystem 2, again, you're going to have light striking the photosystem. An electron gets excited. That electron is going to again travel down an electron transport chain. That electron can then replace the electron that was lost in photosystem 1. Now, as this electron is traveling down this electron transport chain, it provides the energy for chemiosmotic synthesis of ATP, which is going to be important because it's needed in order for the dark reaction or the Calvin cycle to occur. Now, how are we going to replace that electron that was lost in photosystem 2? Well, the splitting of water is going to enable us to replace that electron that was lost. So the splitting of water is going to release oxygen and it's going to release hydrogen. This oxygen is going to leave through the stomates, or it's going to be used for cellular respiration. Our hydrogen, remember, hydrogen consists of a proton and an electron. So what this proton is going to do is that proton is what's going to set up a proton gradient, and that's what is going to be formed in the thylakoid space, is that proton gradient. Now, as far as where is this electron going, well, ultimately, that electron replaces the electron lost in P680. It gets sent along an electron transport chain. It ends up in P700, And then, right, so it replaces the one lost in P680, goes down an electron transport chain, ends up in photosystem 1 in P700, gets excited, and ultimately is going to help reduce NADP plus to NADPH. And NADPH is important, similar to why ATP is important, because they're both going to be used for the Calvin cycle. So now let's fill in our note packet. All right, so in our note packets, 1A says photons begin by exciting electrons in photosystem P700. What happens to these excited electrons? So what happens to these excited electrons is that they enter an ETC, an electron transport chain. And what is produced as a result of this electron transport chain, and why is that product important? Well, NADP plus gets reduced to NADPH, which can then be used for the Calvin cycle. So letter C, what molecule ends up with the electrons and becomes reduced as a result? NADP plus. Now, how is this void in P700 filled? It's going to come from the electron in P680, which ultimately came from the splitting of water. Right, so how is the electron void in P680 filled? Because of the splitting of water. Now, what's formed when the water splits is going to be oxygen as well as a hydrogen, which consists of a proton and an electron. That proton is going to be used to set up a proton gradient. The electron is ultimately going to replace the one that was lost in 680, get excited, travel down an ETC, replace the electron lost in P70, get excited, travel down an ETC, and help reduce NADP plus to NADPH. The oxygen is going to get released through the stomates, or it can be used by the mitochondria for cellular respiration. Now, what's produced as a result of this first electron transport chain is ATP, and that's important because that's also needed for the Calvin cycle. So here's non-cyclic electron flow, or non-cyclic photophosphorylation in a nutshell. The low energy electrons originating from water end up in the high energy NADPH, right? The water replaced electrons that were missing in photosystem 2, which ended up becoming excited, entering an electron transport chain. That electron transport chain created energy, which will be used in the Calvin cycle. The electron ended up in photosystem 1, got excited by the photons of light being absorbed, entered an electron transport chain, and then ended up in NADPH. You need to know that ATP is produced as well as NADPH, which are then used in the Calvin cycle to reduce carbon dioxide to sugar. Oxygen is a byproduct. Most of it escapes through the stomata. Some of it can be used for cellular respiration. And this type of photophosphorylation, non-cyclic photophosphorylation, involves two photosystems, whereas cyclic electron flow or cyclic photophosphorylation only involves one photosystem.